the chief source of energy for our body. So without it, we could not be able to run or walk. Carbohydrates are polyhydroxyaldehydes or polyhydroxyketones, or any substances that yield these organic compounds. These carbohydrates are categorized mainly into three: the monosaccharide, the disaccharide, and polysaccharides. So like this cake. This cake is the best source of carbohydrates wherein it is made up of wheat, which is a starch, that are considered as polysaccharides, and of sugar that can be found in chocolate and as an additional ingredient for this cake.
Proteins are high molecular mass polymers containing the elements of carbon, hydrogen, oxygen, and nitrogen. Proteins make major instructional and functional contributions to the cell and to the organism as a whole. Because of the continual use and degradation of the system, protein must be synthesized almost constantly. This stage, let's explore another macromolecule, which was the nucleic acid. What we are taking today to know this nucleic acid and how it will affect to our acid.
Lipids, the stored energy in our body, have been used as insulator, shock absorber, and it is part of many metabolic processes. Lipids yield fatty acids, a hydrolysis or combined with fatty acids to form ethers. It is insoluble in water but soluble in organic compounds. Lipids can be classified into three types: simple, complex, and derived. So store margarine is an example of lipids.
For this day, let's explore another macromolecule, which is the nucleic acid. What we are aiming today is to know this nucleic acid and how it will affect our daily living.
DNA is primarily responsible for the transfer of genetic information. The Watson crick made of DNA. The chains are composed of alternating sugar and phosphates, with the sugar being bonded to one of the four different heterocyclic compounds: the adenine, the guanine, cytosine, and thymine. That's the reason why we can be able to get the features from our father and mother. Nucleic acids are carriers of hereditary information itself. These are macromolecules, huge polymers with molecular masses of over 100 units. There are types of nucleic acid: the deoxyribonucleic acid or DNA and the ribonucleic acid or RNA. RNA is primarily responsible for protein synthesis. There are four kinds of RNA: ribosomal RNA, messenger RNA, transfer RNA, and heterogeneous RNA. So first, ribosomal RNA. Ribosomal RNA is a major function of the total RNA that combines with protein to form the ribosomes. Next is messenger RNA. It is concerned with the transmission of genetic information from the DNA to the site of protein synthesis. Third, transfer RNA holds a specific amino acid for incorporation into a protein molecule. And last is heterogeneous RNA, a precursor of mRNA and can be found in the nucleus of the cell. We are done knowing the four types of RNA. So now, let's check the video to make you understand more about the function of the four kinds of RNA in the process of DNA and RNA.
The cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA. An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides. Both DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the four bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA. In their final form, these molecules play a vital role in protein synthesis.
The process of DNA replication, translation, and transcription are really amazing. Yet, it was complicated. If this process would not be properly done, it leads to genetic diseases. Genetic diseases can be classified into two major categories. First is chromosomal disorders. Second is the monogenic disorders. So first is chromosomal disorders, in which there is an excess or loss of chromosomes, deletion of part of a chromosome, or translocation of a chromosome. Examples include Down syndrome and chronic myelogenous leukemia. The second is monogenic disorders, which involve one mutant gene. It is further subdivided into three: autosomal dominant, autosomal recessive, and X-linked. So example of autosomal dominant is Huntington's chorea. For autosomal recessive is cystic fibrosis, and X-linked is hemophilia. There are methods can be used in the treatment of genetic diseases. These are: correct the metabolic consequences of the diseases by supplying the missing product. Second is replace the missing enzyme or hormone. Next is remove excess stored substance. And last is correct the major genetic abnormality.
I threw a wish in the well. Don't ask me, I'll never tell. I looked at you as it fell, and now you're in my way. I trade my soul for a wish, pennies and dimes for a kiss. I wasn't looking for this, but now you're in my way. Your stare was holding, ripped jeans, skin was showing. Hot night, wind was blowing. Where you think you're going, baby? Hey, I just met you, and this is crazy, but here's my number, so call me maybe. It's hard to look right at you, baby, but here's my number, so call me maybe. Hey, I just met you, and this is crazy, but here's my number, so call me maybe. And all the other boys try to chase me, but here's my number, so call me maybe. You took your time with